Okay, good afternoon, everyone. Welcome to the Bio-Trac weekly webinars. My name is Mark Nardone. I am director and co-founder of Bio-Trac. Welcome again, like I said, to our webinar series. I ask that the questions that you may have will be submitted into the Q&A box, which is towards the bottom of the page, and we will address those questions as time permits at the end of the presentation. For those who might not be familiar with Bio-Trac, Bio-Trac offers hands-on training workshops for research scientists. These workshops, which are team taught, focus on the latest relevant techniques that are necessary for laboratory research. We were originally based at the National Institutes of Health for 30 years, and Bio-Trac is now located on the Montgomery College Germantown campus in the Bioscience Education Center, and these workshops are open to the scientific community. Since we started this program, we have provided hands-on training to over 17,000 scientists. And it's my pleasure today to we have an exciting announcement. We are going to be offering a series of live remote training workshops this summer, starting with our gene editing with CRISPR workshop in July on the 15th and the 17th, and also a single-cell RNA-seq workshop on July 27th through the 30th. The CRISPR workshop will be directed by Dr. Sergey Kozlov, who's with NIH, NCI, and again, team taught. And the single-cell RNA-seq workshop is directed by Dr. Michael Kelly, who's the director of the single-cell core at the NIH with NCI, as well as Dr. Sejun Young, who's also at the National Institutes of Health. All of these remote workshops will be team taught. They'll have live lecture and laboratory demonstrations, as well as protocol review and discussion. All participants will get copies of all presentations given during the program, reference material, lab protocols, everything you've come to expect from attending one of our workshops. Today, I'd like to go ahead and introduce to you Dr. Maria Hernandez. Dr. Hernandez is a research scientist with Frederick National Laboratory in the recently established NCI single-cell analysis facility. She was previously a research fellow at the NCI CCR, where she applied single-cell RNA-seq to study the transcriptomic landscape of tumor cell communities in a hepatocellular carcinoma and to explore the impact of tumor transcriptomic diversity on patient outcome. She has experience in single-cell analysis methods and sample processing. So without further pause, I'd like to introduce Dr. Maria Hernandez and her presentation on sample processing considerations for single-cell sequencing. Maria, they're all yours.
Thank you, Mark, and hello everyone, and welcome. Today I would like to talk to you about sample processing for performing single-cell RNA-seq experiments. And then while sample processing seems to be easy, it is often overlooked, and sometimes it is performed under one standard protocol that fits all. But the reality is that proper single-cell suspension preparation is vital for acquiring single-cell RNA-seq data. So I would say that it's worth investing the time and effort in the sample preparation step. So I would like to start off by briefly going over the basics of single-cell RNA-seq. Then I'll move on to discuss some considerations for the sample preparation and tissue association. Then I'll end up by going over some effects of some sample preps reflected in the final sequencing data. And finally, I'll provide some helpful links and the conclusion. So by now, single cell has become a well-adopted approach to analyze tumors and study their heterogeneity in their cell populations. Here is a comparison between bulk RNA-seq, which we are more familiar with, and the single-cell RNA-seq, which is becoming more popular by now. Bulk RNA-seq measures tissue gene expression levels that are average expression profiles of individual cells within a tissue. By comparison, single-cell RNA-seq preserves the information about the cell of origin for each RNA molecule that is being measured. So here is a general workflow in single cell. It involves, obviously, the dissociation, whether if it is from a solid tissue or a liquid sample, like PBMCs. The idea is that you would want to dissociate the sample into a high viable single cell suspension. Depending on the experimental design, sample preparation involves some sorting for selecting specific populations of interest. The next step, once you have your single cell suspension, it's followed by the droplet encapsulation or the partitioning of cells. This is followed by transforming the mRNA into cDNA, then amplify the cDNA and prepare the libraries for sequencing. And then obviously, after you sequence, the last part will be the data with the identification of the transcripts that are expressed by each cell being profiled. But today, I'm just gonna focus in this first part of the process, and let's dig into it. So here are some general questions that you should be asking before processing the sample and actually before you design the experiment. So first you may wanna ask, what tissue type am I gonna process? Is it a solid tissue or is it a liquid sample? Now, is that tissue fresh or is it frozen? Then you may wanna move on and ask, how many cells will I need to accomplish the question that I wanna answer? And how much tissue do I need to process to get that number of cells desired? So for that, you may wanna consider the platform you wanna use to perform your single cell experiment. For instance, if you're doing single cell SmartSeq, you may wanna consider how many plates you're gonna need to have to process the cells. Or if you're doing any droplet based, you may wanna know how many cells you wanna target for the capture, let's say in 10x genomics. So for instance, 10x genomics recommends loading at least 90% viable cells or higher. And it is often, when you don't have a viable sample and you have a lot of dead cells, it is often the underlying cause of below number of target cells, and then it can contribute to the background signal or ambient RNA. So then for that, I would say it's very important to have a really rich viable suspension. With that, you wanna make sure that you don't have a lot of red blood cells present. That might also interfere in your final data. For that, you would wanna go and do red blood cell lysis. And then if you're interested in a particular cell population, this is when you will go and do enrichment. Or if you are interested in just analyze any type of cell embedded in the tissue, then you just avoid the enrichment and just focus on the viability of the suspension. So let's dig a little bit more into each of these questions before moving on. So let's talk about tissue processing. Fresh is best, and it's actually the gold standard. However, there are many instances in which access to fresh samples is virtually impossible. Therefore, we have to work with what we have, and we have to pay close attention on how that particular tissue was procured, how was it stored, and how was transported to you. So if you have access like we do at NIH, that we're working closely with the clinical center, which is literally across the street, we will coordinate in advance to be able to provide an appropriate storage buffer or fresh media into the OR or whoever is going to be transporting the sample to you. I would recommend that you would keep that sample at 4 degrees. Also, it is crucial to minimize the ischemic time. I personally prefer to be able to start processing a fresh sample within 20 minutes of its collection. But as I said, not everyone has access on super fresh samples, especially if it's human derived, but you have to work with what you have. Now, if you have the sample, let's say fresh in this case, you may want to ask if the tissue you have can be effectively dissociated. It is quite different to process liver tissue versus brain tissue. So for that, you have to consider how is the way that you're gonna dissociate. So now there's several options once you have fresh tissue. The most common one, and the one that I'll be focusing on, is the mechanical and enzymatic dissociation approaches. There is also the possibility of doing dissociation with cold active protease. Or if you cannot do the capture that day, there's the option of cryopreserve the sample. By that, I mean that once you get the fresh tissue, you may want to dissociate upon arrival and then cryopreserve in freezing media and store in -80 and later on in liquid nitrogen. Now there's a caveat with that. You have to account that upon thawing your cryopreserved sample, you may lose 40% of your viable cells. Now another possible option is methanol fixation. And then finally, and the one that I'm gonna also discuss here, for some of the tissues that have been previously procured or are being flash frozen, there's the option of single nuclei isolation. So let's go over the most common one, which is the mechanical and enzymatic dissociation. So this is a very simple and informative flow chart that was just published in a paper from last month from Nature. This flow chart focuses on fresh samples. So some things to consider is that for dissociation, you may want to select mixtures, enzymatic mixtures based on the specific characteristic of each tumor type, such as cell type composition, if you know them, or extracellular matrix components. For example, liberase is used to break down collagen fibers in breast cancer tissues, whereas to break down extracellular matrices in glioblastoma, you may want to use papain, which is a cysteine protease. It is also recommended to include DNase 1 to digest any possible DNA that is being released by dead cells. This may decrease the viscosity in all dissociation mixtures and helps to eliminate undesired background noise. So here, I'm also including what we currently we use when you don't know what enzymes specific are most useful for your type of tissue. You may want to consider using the Miltenyi tissue or tumor dissociation kit. It works well across different types of tissue, but if you have the time and the tissues to optimize up better dissociation, I would recommend to test other specific enzymes. And then at the end, you just really have to take into consideration if any of these enzymes may affect the sample preparation and later on your RNA-seq data. So to recover the expected number of cells, it is critical to maximize viability and minimize the cell preparation time. So dead cells can be eliminated by using dead cell removal kit. That's what the one that we use in the lab. With this kit, you label cells with the dead cell removal microbits and load them over an LS column in the magnetic field of max separator. For instance, 10X recommends, as I said, that the cell suspensions that have viability below 70% or 80% should go through the dead cell removal step. You have to take into account that when you do that cell removal step, it may result in considerable cell loss. I would say that if you have at least 2 million cells to start with, it would be worth doing that cell removal. One thing that it's not a major effect, but might improve the loss of those precious cells would be doing low spin washes. Because as many of you are familiar, at least for 10X, there are 3 different washes performed after you have your single cell suspension, and this is before loading the microfluidic device. Now, another thing that is optional, but people consider is that you want to enrich your cell population for a specific type of cell that you want to target. For that, the most common approach is to use magnetic labeling and then magnetic separation um, using the Miltenyi kits. And there are many commercially available kits to target specific population. One option, for example, and this is something that I have done, is that if your interest is in specific immune cells, it is advisable to first enrich your population by depleting CD45 cells first, and this might lead to a purer immune cell preparation. Something else to keep in mind, people also use FACS for enriching populations. FACS, well, is very useful. It might introduce additional stress, and that has to take stress to the cells, and that has to take into consideration. But regardless of any process, any manipulation or enrichment of cells will have an effect on the downstream data. And you need to know the process to keep in mind when you are interpreting the final sequencing data. So there are some studies that have shown that enzymatic digestion of tissues can induce gene expression artifacts that can affect the single cell RNA-seq data interpretation. It has also been shown that the effects depend not only on the enzymes, but also on the temperature and the duration of the digestion process. I mean, you might expect that different cell types have different susceptibilities to this type of stress. In this recent study from, actually this paper came out last month, different enzymatic dissociation protocols were compared in parallel. So this was for a non-small cell lung carcinoma tissue. And then 3 different protocols were tested. For instance, the C4 protocol only involved liberase, whereas the C4 protocol only involved using DNase 1 and collagenase 4, whereas the LE protocol involved only liberase. The PDEC protocol, it's involve a mixture of more enzymes that include pronase, elastase, dispase, among others. So at the bottom of the figure, this is the UMAD plot. At the bottom of the figure, they show the proportion of cells in each subset, the proportion of cells that are being profiled or that end up being showed upon dissociation using these 3 different protocols. So K is the number of cells, and then the colors depict the cell type that it's being detected. Another way to look at the protocol variation is by looking at the median distribution of the number of reads per cell and the number of genes, and the median distribution of the number of genes per cell expressed. So as I said previously, ideally, the input cell suspension into the microfluidic device should contain more than 90% viable cells. Non-viable and dying cells generally contain less and more fragmented RNA that may not be efficiently captured by 10x. In the presence of a high fraction of non-viable cells in the input suspension may therefore decrease the efficiency of the partition and recovery. Here is a picture in which there's pre-dead cell removal and post-dead cell removal. We use acridine orange to detect the green dots, the live cells, which are the green dots. And so here you can clearly see the combination of a bunch of dead cells. And so it produces like, it means that there's only like almost 60% viable. So you can see that there's a lot of dead cells. And then that is being reflected, let's say, on the final data. There's, when you look at the UMAP, you don't see a clear inflection point into the distribution of cells versus background. And then here is the TSNE plot that reflects not having the dead cell removal. Now, when you process your sample through dead cell removal, you significantly increase your viability into 95% at least, or even more. And then here is reflected in the pretty nice knee plot, in which you clearly see the inflection, in which you can differentiate the cells versus the background. And also, it is very important to accurately count the number of cells before loading the cell suspension into the microfluidic device. Overloading those devices or those chips can induce clogging and increase the rates of doublets in your final data. So as I said, not processing or mechanical digestion of a fresh sample is not possible. And let's say you have a frozen sample, there's an option, and that option is the single nuclei RNA-seq. I would say that single nuclei involves a more robust protocol optimization, and it's not trivial. It's very laborious, and but it's a good option for not only for frozen tissues, but also for difficult tissues to access. So many people are not convinced or want to be convinced that analyzing single nuclei, it is the same or is comparable to a single cell. Well, there's a study here in which showed there is a pretty good correlation, pretty decent correlation between profiles from single cells and single nuclei. So in this study, single cell RNA-seq and single nuclei RNA-seq derived from mouse brains were performed in parallel, showing this shows a pretty nice correlation among both. So the only disadvantage of, sorry for the background noise, my kids fighting downstairs. So I would say that the only disadvantage of single nuclei is that there is a low gene, it could be a low gene detection rate with a larger amount of intron retention. Likely that comes from pre-mRNA. I have to point out that single nuclei is actually required for ATAC protocols, and it will be required for the upcoming combined single nuclei and gene expression, single and ATAC and gene expression protocols. And so finally, I mean, you wanna take into consideration that if your sample is limited or precious, consider the implications of possible sample loss, as this optimization it's not trivial. So just to wrap up, in conclusion, I hope the take-home message is that a successful single-cell study starts with a thoughtful consideration on how to prepare the cell or nuclei suspension. And there are many choices to prepare the sample, but those are related to the tissue type, the study design, the question you want to answer. So you really have to lay out your experimental flow chart beforehand. And gene expression artifacts are always, or most of the time, going to be present in your final data. So be wary of those and be able to identify them. So here are some useful links. 10x Genomics has a pretty good set of videos that demonstrates protocols and explains how the technology works. Miltenyi Biotech also has a good set of videos. And for those of you that belong to the NIH community, there's the single cell genomics interest group. And then I wanted to add these these two newest articles. One is from Nature from last month, and actually this other one, this other one, it's from, came out last week, I think. Another good resource to look at specific question regarding association is this book, The Single Cell Methods. And with that, I would like to welcome any questions. This is my group. Mike Kelly is a lead scientist in SCAF. We are located, although we belong to Frederick National Lab, we are located on the NIH main campus in Bethesda, literally or pretty close across to the clinical center. And this is the group I work with. So it's Mike, and then there's Kimia, who works in the bioinformatics, and Allison, who's the guru of capture, 10x captures, and library preparation. So with that, I would like to welcome some questions.
Maria, outstanding presentation. Thank you so much.
Thank you, Mark.
All right, we got a couple here. What quality control assays are recommended for sample prep other than viability assays?
One that it's very useful. I mean, again, you want to minimize the time of processing, so there's not much time that you have in hand to do. But I would look at the like count the samples, like take an aliquot in every step, take an aliquot of your sample, and then actually you can look in the microscope and see how the dissociation is going. We typically do that, especially if we want to see before the washes, how much debris do you have. That's the only one that I would say it's important.
Very good. Next question, does 10x system work for fixed tissues or cells?
No, I mean, not now, at least on the device. I'm not aware, and we cannot load fixed samples. That's the downside. But you can do nuclei extraction, and then.
Next question, any special advice or considerations for single-cell RNA-seq of skin? This is one of the toughest tissues for single-cell RNA-seq studies.
Yeah, it is. I actually came across about a paper that laid out how to prepare skin tissues. I have not personally worked with that type of tissue, but if Mark, if you can give me the information or the email of the person that is asking, I can send out the paper that I had read. I don't have it with me right now, but I'm pretty sure I saved it, so I can share that with that person.
No, outstanding. Actually, send me the link and I'll send it to the entire group for others.
Absolutely.
Who didn't ask the question. See, next question. How many years frozen tissues can be used for single nuclei seq?
I mean, there's not a specific time frame. I would say that if you have the option of using the most recent ones, that would be ideal. I mean, it's like a chain of events. You really don't know how those tissues were collected and how those tissues were preserved. So I wouldn't go beyond 5 years, and I think I'm extending it for, I mean, I think even 5 years is too long. It's impossible to know unless you can test. I mean, if you can afford to prep some of those old tissues and at least look you look on the microscope, the nuclei, how it looks, but it doesn't guarantee that those nuclei might have, reliable RNA. So that's the caveat with nuclei, that there's no QC step up until you sequence the data. So no, there's no one answer for that, sorry.
Alrighty, next, any experience or advice on cold digestion enzymes?
With cold protease, I have not used it. There's a few papers out there that seem to work. I think if I'm not wrong, that particular protocol of using called active protease, it's very useful in, I think it was in skin cells or in kidney cells, but I can send also that reference to you.
Okay. We have about another 10 questions. So what I'm going to suggest is if those who are submitting questions, email them to me. My email is mark, M-A-R-K, Nardone, Nardone, at Bio-Trac, Biotrac.com. And I'll go ahead and put it in the chat in a second. And I'll go ahead and send them to Maria, and we can answer them to the group. So before we log out and such, let me put my email address in the chat so you have it.
And I would say that, I mean, if some of you are in NIH, I mean, SCAF, our facility is pretty open. Like we're not a regular core facility. We also advise people on what steps to take if they have a specific question and they want to utilize these single cell technologies. So feel free to email any of us at SCAF. We're pretty easy to find in the CCR, I guess, website. I can also, send my email. I have not had experience with every type of tissue, but at least I can refer you to someone or to some paper that have done.
Maria, wonderful presentation, and thank you very much for the timely references, also outstanding. I'd like to thank everyone for attending today's event. Next Tuesday, you will receive an email from my office containing a link to today's recording of the PowerPoint, of the presentation, as well as a PDF of today's presentation. Next week's webinar will be given by Dr. Michael Sirachi from AstraZeneca, focusing on multiplex immunofluorescence staining imaging and image analysis. While we're talking about multiplexing, our third annual AstraZeneca Bio-Trac Immuno-Oncology Immunofluorescence Multiplexing Symposium and Workshop has been postponed. It was originally scheduled for October. It will be March 9th to the 12th, 2021. If the March event is anything like what we did last year, there'll be over 100 scientists from around the world in attendance. It's a phenomenal event. As I mentioned earlier, we are going to be doing live remote training sessions starting in July. In July, we'll have CRISPR single-cell RNA-seq again with Michael Kelly and his group. We're also going to be doing a R for research scientists in August, a flow cytometry, principle and methods. And again, all these workshops will be live. There'll be lecture, laboratory demonstration, protocol review, discussion. We're excited about doing this. I know we took a little bit of time just to make sure we had everything in place before we go ahead and put these things forward to the scientific community. Again, thank you for your time, and we'll be talking with you shortly. Take care.
Thank you.